Welcome back to BioB 160, Principles of Living Systems. I'm Ruth Reitzman, your instructor for this course. And in this episode, we'll explore the structure and function of cell membranes. Let's get started. All cells have a plasma or cell membrane. The plasma membrane is a very important structure that serves as a boundary and separates the inside of the cell from the outside of the cell. The plasma membrane regulates what enters and exits the cell through its selective permeability. In this episode, we'll answer these questions. What is the plasma membrane made of, and how do substances move across the membrane? The plasma membrane is a fluid mosaic with a fluid sea of phospholipids floating, and floating in the sea are the proteins. The proteins can move around the membrane, somewhat like icebergs floating in the ocean. This is a diagram of the plasma membrane showing the phospholipid bilayer and the proteins embedded in the membrane. Notice the hydrophilic or water-loving regions of the proteins point to the outside and inside of the bilayer, while the hydrophobic regions of the proteins are embedded in the bilayer. Membranes vary in their fluidity. If an organism lives in cold temperatures, like a fish living in the Arctic, its membranes will have more unsaturated fats to keep the membranes fluid. Organisms living in warmer temperatures, or organisms that regulate their body temperature, such as mammals, have more saturated fats, which pack together. In animal membranes, cholesterol is also added to give the membrane strength. If we look at the outside of the plasma membrane in animal cells, we see that there are many proteins attached to the outer surface that make up the extracellular matrix. These proteins often have carbohydrates attached and are called glycoproteins. The inside of the membrane is also attached to proteins. Some of these proteins are part of the cell's cytoskeleton. The cytoskeletal protein often help anchor the membrane proteins. Membrane proteins that span the entire width of the membrane are called integral membrane proteins or transmembrane proteins. This diagram shows a ribbon model of a transmembrane protein. Many of these proteins have extensive alpha helical regions that fold together and form a channel or pore through the membrane. Let's look at some of the functions of membrane proteins. Membrane proteins may transport specific molecules into or out of the cell. Some membrane proteins have specific enzymatic activity, and other membrane proteins help relay information from the outside of the cell to the inside. This signal relay process is called signal transduction. Some membrane proteins bind to the membrane proteins of other cells, allowing cells to recognize each other. An example of this cell-cell recognition occurs in the immune response to foreign cells, such as pathogenic bacteria. Some proteins are involved in cell joining, such as the proteins that form gap junctions or tight junctions between cells. And some membrane proteins attach to the extracellular matrix and join cells together to form tissues. So now that we've looked at membrane structure and function, let's discuss how substances cross membranes. The cell membrane is a selectively permeable barrier. There are two ways for substances to enter the cell, through the phospholipid bilayer or through the membrane proteins. The phospholipid bilayer is a barrier to most molecules. Only very small, nonpolar molecules pass easily through the lipid bilayer. Examples of these are oxygen, O2, or carbon dioxide, CO2. Most substances are larger or are polar, so they will only enter if specific proteins can act as a gate or entry point. In terms of energy requirements, there are two categories of transport: passive transport, where no energy input is required, and active transport, where energy is required. Diffusion is a type of passive transport. If you place a drop of red food coloring in a beaker of water, the molecules of dye will spread out and the drop will eventually be evenly distributed throughout the beaker. This is an example of diffusion. Diffusion is a passive process since no energy input is needed. The molecules of dye will randomly move through the beaker until they are evenly distributed. Small nonpolar molecules that can move directly through the lipid bilayer move by diffusion. This is also called simple diffusion. This diagram shows the movement of a small molecule across the membrane from a high concentration to a low concentration until an equilibrium is reached, or as there's no net movement of molecules across the membranes. Osmosis is the diffusion of water across a membrane. Cells are filled with many different molecules, and most of these molecules are polar or charged. Polar or charged molecules do not move easily through a lipid bilayer. Water is also a polar molecule, but since it is small, it does move through the bilayer. It is also assisted through special protein channels called aquaporins. Now let's consider different conditions of salt solutions. Since salt ions are charged, they don't cross the lipid bilayer. If cells are placed in a solution which has a salt concentration equal to the salt concentration inside the cell, the solution is said to be isotonic, iso for the same, and the water molecules will have no net movement in or outside of the cell. This is shown in the middle panel of the diagram. Cells in an isotonic solution will remain their normal size and shape. If a cell is placed in a very dilute solution with very little salt, the water from the outside of the cell will tend to move inward. This condition is shown on the left panel and is called hypotonic. Cells placed in hypotonic solutions will increase in size as the water from the outside of the cell moves in. In animal cells, the cells may lyse or burst due to the excess water pressure inside the cell. Plant cells are protected from hypotonic solutions since their cell walls can resist the added pressure. If a cell is placed in a very salty or hypertonic solution, the water will move out of the cell and the cell will shrink. This will affect both animal and plant cells and is called plasmolysis. Now that we've looked at the movement through the lipid bilayer, let's look at movement through protein carriers. The type of movement shown here is facilitated diffusion. This is movement through a channel or carrier protein, and it moves molecules from high concentration to low concentration. Since the movement is from high to low, this is a type of passive process. The other type of transport that requires a protein is active transport. Active transport moves molecules from lower concentrations to higher concentrations. Since this is a kind of uphill movement, it requires the input of energy. So active transport usually requires energy in the form of ATP, adenosine triphosphate. The example shown here is the sodium-potassium pump that is present in nearly every cell of our body. The sodium-potassium pump moves 3 atoms of sodium out of the cell and moves 2 atoms of potassium into the cell. This slide reviews the 3 methods of the transport that we've talked about so far. Both diffusion and facilitated diffusion are types of passive transport, and they don't require an input of energy. Diffusion, or simple diffusion, moves directly through the lipid bilayer, while facilitated diffusion moves through protein carriers. Active transport requires input of energy and also involves a carrier protein. Now let's look at how large molecules cross into cells. Very large molecules, such as proteins or small food particles, are too large to cross through the membrane proteins. They can enter into the cell when a cell pinches off part of the membrane and brings the particles into the cell in a membrane vesicle. Endocytosis is the entry of particles, and exocytosis is the exit of particles. There are several types of endocytosis depending on the amount of membrane engulfed and if specific receptors are needed. Phagocytosis, or cellular eating, involves a large region of membrane and is used by protozoa to engulf food particles. As we saw in Chapter 6, phagocytosis is used by our white blood cells to engulf bacteria so that they can be digested by our lysosomes. Pinocytosis involves a smaller piece of membrane and forms small vesicles. This is sometimes called cellular drinking since it allows cells to bring in small molecules and fluids. A third type of endocytosis is receptor-mediated endocytosis. In this type of endocytosis, specific receptors in the cell surface bind to specific molecules called ligands. The specific receptor-ligand binding causes the cell to form a vesicle, which pinches off and brings the molecules into the cell. After this episode, you should now be able to describe and identify the components of the plasma membrane. You should also be able to distinguish between the following terms or sets of terms: peripheral and integral membrane proteins, diffusion, osmosis, facilitated diffusion, and active transport, hypertonic, hypotonic, and isotonic, and endocytosis and exocytosis. This concludes this episode of BioB 160. To continue your study, be sure to do the homework on the MyLabs Plus Mastering Biology site and post to this week's discussion board. And don't forget to watch the great animations on the cell membrane, including the bioflicks on membrane transport, in the Chapter 7 resources for MyLabs Plus. Thanks and have a great day.